This is for Biology 223. This is discussing the paper Evolution in Flower Color Pattern through Selection of Regulatory Small RNAs. This is part 2 where we had started a problem. I was just setting it up. The problem would be, imagine we have in this biosynthetic pathway controlling color patterns, a situation where we have a heterozygous individual that's heterozygous at all these loci involved, crossed to itself or another of the same genetic background here. And we can ask what proportion would be, let me see, I did the problem here, what proportion would be able to create yellow pigment, but yellow only yellow pigment. So if we think about that, in order to be yellow, to produce this yellow, it then has to have activity of CHS. So if we do the typical Punnett square, I'm going to show that here, right? We can have functional and null, and we expect that the product is going to be the homozygous wild, homozygous functional, the two heterozygous, so 1 to 2 to 1. So we're going to have this 1 to 2 to 1 ratio, and if we consider that the heterozygous is functional, produces sufficient gene product to function completely, which might be typical here. We expect that. We expect white flowers or red flowers, not pink flowers, although codominance can occur and partial dominance. But here, the typical thing is that. And so we're going to have 3/4 have a functional and 1/4 have a non-functional phenotype. That is gene product, sufficient gene product versus not. So in order to get yellow, what do we need? We need CHS. We have to proceed past that. So 3/4 will proceed past that. Oops. So let me, I'll do it here. 3/4 have to proceed past that. And then we have to not proceed past, not have any CH1 gene. So it's that. CGT has to be functional in order to get yellow, so that's going to be 3/4. But sulf doesn't work, it can't work, so it has to be 1/4. And AS1 has to work, so that's 3/4. So 4 times 4 times 4 times 4 times 4, that's 2 to the 5th, that's 1024. So let's do that in 1024. So yellow only, we're going to have of 1024, we can do the whole problem with 1024. 3 times 3 times 3, that's 27. So we're going to have 3 cubed, which is going to equal to 27 1024. Okay, so yellow, what other phenotypes are possible? That's yellow. We can imagine, let's see, what's the next one? The next easiest one is yellow and purple, right? It can make yellow and purple pigments, but we're not including things that must be white. And you might think here, in the real flower, well, the whole petal, all the cells in the petal have the same genotype, so how do we get a pattern? And of course, there are other regulatory mechanisms that control the expression of these genes or the biochemical activity of their gene products by biochemical mechanisms. So we can have genetic mechanisms or biochemical mechanisms superimposed upon this. So what we're looking at here, the genotype, is what is actually possible to make. So to be yellow plus purple, it has to have active here, active here, active here, not active here, active here. So that's not much different. So it's going to be, so let's do it here. So we have to have likewise a functional CHS, and now we have to have functional CH1 to make the purple, rather non-functional, and the CGT also must be functional, the sulf also must not be functional, and the ASI must be functional. So we get something quite similar. It's 4 to the 5th, so it's 10, it's out of a 1024 again. And here it's going to be 3 times 3 times 3 times 3, so that's 3 to the power of 4, so that's 81, right? 9 times 9 equals to 81 1024. Okay, that's also quite straightforward. Now it gets a little bit complicated, and I'm not sure I'll do all possibilities, but let's imagine what's the answer to something that can be purple, but no yellow, no yellow pigment produced. So that's a little bit trickier. because it can have here, okay, to make purple, it has to have CHS. So let's do that. That's fine. It has to have CHI, right? That's fine. So that's, sorry, 3/4. It doesn't have to have anything else. It only has to have purple, no yellow. So it only has to make that. So you say, well, that's easy. It doesn't have this, right? So we say, well, it's 1/4 of CGT, and it stops there. Yes, but it could have CGT and also have sulf. So it could have non-functional CGT, or it could have functional CGT. So it could have non-functional CGT, and it doesn't matter whether sulf or AS1 works. So that's that. So it could have non-functional, and it doesn't matter about those. Or it could be it's functional at CGT, but sulf is functional, right? If it's goes through here and this gene product works, but this one doesn't, then that'll also be white, and then it doesn't matter what's here. Or it could be that it has functional CGT, non-functional sulf, and non-functional AS1, and that will also work. So if we add that up, it's going to be 3/4 times 3/4 times this plus this plus that. So we're going to get this. We're going to say here we have 3/4 times 3/4 plus, times the sum of what? Well, 1/4 times 4/4, and I can do that times 4/4 times 4. So why do I want to do it? Why am I putting the 1 as 4/4? To keep everything in the common denominator. Plus, so it's going to be this times this times these, this times this times these. So 3/4 times 3/4 times 4/4 plus this one, the 3/4 times 1/4 times 1/4. Sorry, I'm running out of room here. Oops, can't quite see that. Can we see that? We can't see that. I'm sorry. Let me do that. I think you can see that, but I can't see that. You can see it as it goes. Plus 1/4 times 3/4 times 1/4 times 1/4. And so if we do that, we're going to say, okay, this is all going to be 10/24, 10/24, and 10/24. So what we're going to get is we're going to get out of 10/24, we're going to get 3 times 3 times 3 times here 16 plus 3 times 3 times 4, so that's 36 plus 3, right? So what is that? That is 39, that is 55, and 55 times 9, I used a calculator here, that's 495, you can figure that out. 55 times 9, that's close to 55 times 10, that's 550 minus one of those 55s, so we end up with 495. So 495 over 10/24. So why am I keeping track of that? So that's the number for purple. So we've done yellow only, yellow and purple, purple only, and I'm not going to go through here. I leave it to the student, the question of no yellow, no purple, and I think that's the hardest. And I'll tell you the answer here because I think it's obvious. So you could take a shortcut, which is obvious here, is it's going to be one subtracting the other because that represents the totality, right? The white, yellow, and purple, the white plus the yellow and purple is going to be 1. So you could do the short trick here just to say it's unity subtracting this, and that works, but it's a good exercise when you don't know the answer and you don't know whether these numbers are correct, to go through that, and you will see that you're going to need to have something similar to this. You're going to have this, right? What is it going to be? It could get stopped here, and if it's not stopped here, it needs to be stopped there, and if it's not stopped here, it needs to be stopped here, and it needs to be stopped here, either here or by this or here. And so you get something where you have three terms, and that ends up to be 421 10/24 for white, or unable to make purple, no yellow, no purple, and it adds up, and you can see that they all add up together to 10/24 10/24. So that's a good exercise. And how complicated can these be? Well, they can be quite complicated, and you can imagine you had the homework example of Hymenoptera where the sex determination is haploid diploid. That's not really that complicated. That's something very similar to male, female mammals of the X chromosome. What other complications can we have? We can have a linkage, we can have more branches, we can have other things. Linkage is the most complication. I'm going to stop here.